I'm John, a doctoral candidate studying cancer biology, and today, in 10 sentences, we will learn about the human cell from its outer plasma membrane to energy production in the mitochondria, DNA storage in the nucleus, protein synthesis in the cytoplasm, and the intricate processing of proteins in the endoplasmic reticulum and Golgi apparatus. Everyone started life as a single cell, which divided and became approximately 37 trillion cells, which have over 200 discovered cell types, such as oxygen-carrying red blood cells and cancer-hunting natural killer cells. The plasma membrane encases a cell, resembling a water-immersed bubble, primarily composed of phospholipid molecules, which feature a water-attracting half and a water-repelling half. The water-repelling side orients away from water, resulting in a bilayer structure that forms the cell's outer boundary. The plasma membrane allows nutrients and other necessities to be concentrated in the cell, where a series of reactions which do not use oxygen, known as anaerobic metabolism, drive the breakdown of nutrients into energy-rich molecules. The mitochondria, the powerhouse of the cell, a small double membrane structure, uses these energy-rich molecules and oxygen to concentrate protons into a compartment called the intermembrane space. Then, like a hydroelectric dam, channels the resulting proton flow from this overcrowded intermembrane space to a sparsely crowded compartment through a structure called ATP synthase, which stores the energy this flow generates for use by the cell later in a molecule called ATP. Deeper in the cell, another membrane-bound structure called the nucleus holds 2 meters long worth of DNA coiled up like an electrician's nightmare into 46 bundles termed chromosomes, which store essential instructions for a cell's functions. DNA is kept safe from damage in the nucleus, but small copies of sections of DNA, known as messenger RNA, mRNA, carry important information out of the nucleus to complexes known as ribosomes, which use these instructions to make protein-based machines and structural supports for the cell. Ribosomes make these proteins within the empty portion of the cell, known as the cytosol, or they attach to a special protein processing plant, known as the endoplasmic reticulum, which can further control how proteins are assembled with the assistance of differing acidity, alter salt concentrations, such as elevated calcium, and help from protein folding machinery. Not all proteins manage to fold correctly, and those which fail quality testing are tagged with a special molecular marker and destroyed by a massive complex of proteins known as the proteasome. Functioning protein can stay within the cytosol, be released from the endoplasmic reticulum to the cytosol, or travel from the endoplasmic reticulum in tiny balls of membrane called vesicles to the Golgi apparatus. The Golgi apparatus has a 10 out of 10 name choice and also performs further processing of these proteins, then sorts them to be transported by vesicles to their final destination, lysosomes to be digested, the plasma membrane, or secretion to act as signaling or structural proteins in the extracellular matrix, which surrounds most cells and holds them in their proper position. If you enjoyed this journey through cell biology, hit the like button and subscribe. I always welcome comments, including topics you'd like covered in the future, additional factoids, and error correction. Thanks for listening and keep learning.